So I get to talk about my favorite subject, my boys. These are my boys, Charles and Nathan. You know, you never really know where parenthood will take you. It turns out, in my case, it was nothing like I had ever expected. So here they are today. Nathan, who's now 17, is just a great kid, kind and caring. He's about to graduate this coming Saturday, and he's going to go to Texas A&M to study biomedical engineering, and he hopes to become a physician researcher one day. I unfortunately cannot take credit for how he has turned out. It's really all thanks to his big brother, Charles. Charles, who just turned 20 on Sunday, has turned out to be an amazing teacher. He's taught us about acceptance, about compassion, patience, and unconditional love. He has demonstrated incredible courage in the face of adversity, and he has inspired us to do the same. Today, I want to share Charles's story intertwined with the story of citizen scientists working together to make discoveries about a rare genetic condition. Genetics, stemming from Genesis, the origin. So the beginning is always a good place to start, especially since this is a story about a genetics-first approach to curing disease. When Charles was born, my husband and I didn't know that he would take a different developmental path than most children. We couldn't imagine the health challenges that he would face, nor at the time could we even pronounce them. We didn't know that grief would become our steady state, or that we would need to re-engineer our lives to learn to cope with the many devastating diagnoses Charles would eventually receive. Slowly and gradually, Charles's disability began to revealing itself to us. His development in infancy was normal, even early in some areas, like crawling and walking. But at age 2, he was diagnosed with speech delay. At 3, he was diagnosed with ADHD. At 4, he was diagnosed with autism. The diagnosis of autism was very important because it helped us secure those all-important services such as ABA therapy, but it never really helped us make sense of what had happened. The year was 2005, and chromosomal microarray had just become clinically available. What the heck, we thought, what's one more tube of blood? We never expected anything would come of it. We were shocked when we received a phone call from the genetics clinic. The voice on the other end of the call just sounded a little bit too happy. Charles, it turned out, was one of the first patients ever to receive a clinical genetic diagnosis through chromosomal microarray. The condition is called Phelan-McDermid syndrome, also known as the other PMS. In Charles's case, the deletion was very small, deleting a part of a critical gene called SHANK3. In 2005, there was relatively little known about PMS. When we attended our first family conference a year later, we didn't see any other individuals who were like Charles. Prior to chromosomal microarray, most patients diagnosed with PMS had extremely large deletions spanning up to 100 genes. Often their deletions were so large they could be seen under a microscope. Patients with smaller deletions tended to go undiagnosed. Only patients with distinct characteristics suggestive of a genetic condition were even referred for genetic testing. But chromosomal microarray was a real game changer. It could detect extremely small deletions and duplications, enabling the diagnosis of genetic diseases in patients who were previously unlikely to even be referred for testing. Chromosomal microarray became a standard of care for individuals with autism and other forms of developmental disability. As chromosomal microarray became more common, our community size grew, and we began to see patients who were much more mildly affected. We began to realize that PMS was actually a very broad spectrum with a great deal of variability. Through online discussion groups, parents began talking about their children's deletion sizes and trying to figure out exactly what a big or small deletion meant. Did the exact breakpoints matter? What genes were missing? How did deletion size correlate to different aspects of disease, such as whether or not a child might develop speech or experience regression or develop seizures? The information shared by parents was incredible. By sharing with each other, the parents in our community began to learn more about the disease than any medical professional. I want to tell you a little bit more about Charles's story because the work we are doing now is all about patient stories and the power of patient data. In 2007, Charles experienced his first seizure, a generalized tonic-clonic seizure lasting several minutes. A month later, he had another, and then they became a weekly event, and then a daily event. Within 6 months, he was having dozens of seizures every single day. He was diagnosed with a very kind of a rare and refractory form of epilepsy called Lennox-Gastaut syndrome. He suffered from nearly constant injuries from falls resulting from atonic drop seizures. It's a really devastating diagnosis to have. He lost the ability to read and write, and gradually he's been losing the ability to speak. During his worst periods, he would have hundreds of seizures per day. He would be admitted to the hospital where he would stay for up to a month while his medical team tried various approaches to slow down the seizures. The ketogenic diet, the vagus nerve stimulator, countless cocktails of drugs helped for only a few weeks or a few days at a time. You can imagine how scared I was. Charles's future and his life even seemed so tenuous. I was angry, too. It seemed so horribly unfair for a child to suffer so much. And how could we actually know about this gene, SHANK3, and even what it does in the brain, and not be able to use that knowledge to inform the development of targeted therapeutics? Sitting bedside, I made a promise to Charles to do everything in my power I could to help him get better. I talked to the leaders of the Phelan-McDermid Syndrome Foundation, which at that time existed primarily as a support group, about chairing its research support committee. We developed a strategic plan for science, and we prioritized several goals. One of our goals was to tap into the knowledge of parents. As I mentioned before, parents were learning a great deal from each other on online groups such as Facebook. Unfortunately, the knowledge we were gaining in the community was essentially sequestered within our parent group. Without a means of obtaining informed consent, we couldn't use that data for research purposes. And without research and publications, what parents told their children's medical team carried virtually no weight. Every story was important, but we needed a way to gather information in a more systematic fashion. On Facebook, some people posted about everything, others posted about very little. Some topics would generate very lively discussion among many people. Other topics, while important to some people, would generate almost no discussion. Look, Charles's story is not unique. Every day, someone is diagnosed with autism, with epilepsy, or maybe a rare genetic condition. Their stories are similar to Charles's, but maybe not exactly. Making sense of the commonalities and differences is one way we might be able to make discoveries that can help prevent or improve disease conditions. All of us, we all have stories, right? And almost all of us have medical records somewhere. What we lacked in the PMS community was an ability to aggregate all of those experiences and data into a central repository. So in 2011, we launched the Phelan-McDermid Syndrome International Registry. At the time, there weren't very many rare disease groups that had patient registries, so there wasn't exactly a template for creating one. We found a platform called Patient Crossroads, now called Invitae, that hosted our registry. We worked with our research community to develop some questionnaires to capture data about development and health conditions, as well as demographic information. Most importantly, we asked families to upload their genetic reports, and we have genetic counselors who curate the genetic data from the reports and enter it into the patient record. So about 5 years ago, I did what anybody does. I called Zach Kahane, who connected me with one of his colleagues, Paul Aviuk. Together with fellow PMS mom extraordinaire and the principal investigator of our registry, Megan O'Boyle, we submitted an application to PCORI to join PCORnet, a national healthcare network that enables the analysis of healthcare data. Our project, called the Phelan-McDermid Syndrome Data Network, essentially created a Google map of PMS. On top of the genetic data and the parent-reported data from the registry, we layered in data from patients' electronic health records. That was quite a project. Over time, we hope to add in other sources of data, such as data from apps, devices, wearable, omics data, whatever comes along. So what have we learned? We've made some discoveries about how genotype and phenotype are correlated. In this example from a few years ago, you can see that all individuals with PMS, regardless of their deletion size, have abnormal language. That's shown in the yellow. But only patients with the largest deletions have heart defects, shown in blue, or renal defects, shown in green. This helps us begin to answer the question parents have about whether or not deletion size matters and what genes might be involved in various phenotypes in PMS. It's also an important research question, right, if we're trying to develop targeted therapeutics for different aspects of our syndrome. We've also used the registry to partner with researchers who want to explore specific questions by recruiting PMS patients for their studies or simply sharing de-identified data from the registry with them. I should note that of the 1,600 individuals in the world who are part of our foundation, over 1,000 have now participated in our registry. So for those of us, who care about rare disease, it really all starts with hope and the belief that things might just be better. The thing which inspires us to lift ourselves out of a difficult place and persist, even when the going gets tougher than you think you can handle. We live it and breathe it in rare disease. But sometimes all you need is a vehicle for turning hope into reality.